Good morning and welcome to the 2022 CRB Webinar Series. Today's webinar will provide a look at sweet orange scab and citrus canker in California. I am Caitlin Stanton with the Citrus Research Board, and I am joined by Mikhail Machado, who will be running the polls and monitoring the chat, as well as Dr. Joey Mayorkin, who will be introducing our speakers. Please note that this webinar is geared toward growers and pest management professionals. Master gardeners can certainly benefit from participating, but the pest management methods presented, especially the pesticides, should not be followed without a clear understanding of their legal use in home environments. We encourage you to ask questions during the webinar using the Q&A function, which can be found at the bottom of your screen. The speakers will answer all questions following their presentation. And with that, I'm going to pass the mic over to Joey, who will introduce our speakers.
Thank you, Caitlin. It's a pleasure to introduce today's speakers. Please welcome UC Cooperative Extension Subtropical Horticulture Farm Advisor for Riverside and San Diego counties, Sonia Rios. Prior to her current position, Sonia served as a UCCE Staff Research Associate for Tulare and Kings County. As a horticultural farm advisor, Sonia focuses on conducting needs assessments for citrus, avocados, dates, and other commodities to develop and shape programs that serve growers. Currently, Sonia is collaborating with Dr. Gary Bender on a high-density avocado trial to measure yield and its economic advantage, as well as weed management issues in citrus and avocado. Today, Sonia will provide a regional update. Sonia, please go ahead and begin your presentation.
Thank you so much, Joey. And it's really funny because I actually met Joey when he was a graduate student and he helped me a lot while he worked in his lab. Okay, I'm gonna go ahead and share my screen, if that's okay. And where is it at? There it is. So I'm just gonna go fairly quickly on my regional update. But pretty much, we all have seen the same issues in all parts of the state when it comes to labor shortage, of course, the ACP, we're always scared about. However, what I like to focus on just really quickly, just for the fact that I would like to mention that we're also having a couple of seminars is on irrigation. And with the heat coming, we're going to expect a lot of heat waves coming in probably this summer. We've started to get hot already. Kind of scared what August, September might look like. And all I can say is be prepared and make sure that, at least watch the news. I'm sure most of you guys do. And just be on top of what the weather is going to be like and the prediction. I know sometimes even a couple of years ago, we had this massive heat wave that came out of nowhere and caught everybody off guard. Sometimes that does happen. However, you want to bump up your watering schedule just a bit. And if we're going to be predicting a high heat wave, you want to be proactive. And a lot of mistakes that a lot of growers make is that they can actually, okay, we have a heat wave. So the morning of the heat wave, they bump up their irrigation and water and, they think that's going to fix it. Well, that's actually not going to fix it. You need to actually slowly increase the water on your trees so they can slowly prepare themselves and be fully hydrated by the time the heat wave does come, so that it may not be as devastating, hopefully, or because I think in Valley Center they hit like 118 for like 3 days straight, and a couple of growers that were proactive were not impacted because they were doing this procedure. They were actually being proactive and they knew that it was going to be coming, so they started increasing their water slowly. And also for younger trees, you want to make sure you get them whitewashed, or if you just finished pruning or are currently treating something for Phytophthora, you want to make sure you try to get that whitewashed. Heat can be quite damaging. Heat can also affect bloom and fruit set. And if you do have a good amount of fruit, if we start getting more towards the fall and we do have a heat wave there, it can cause fruit split if you are not careful on how you're irrigating. And then just, a couple of quick tips just to remember, you want to check your water lines a lot. You want to actually get out of the truck, you want to walk the lines, you want to flush lines, you want to clear filters, you want to check for leaks, you want to backwash filters. This is also really dependent on the water quality. I would really like to stress this with the people down in San Diego, just for the fact that some parts of northern San Diego, the water quality has declined within the last decade or so. So it's really important that that we backwash those filters. You also want to be on top of injecting acids or chloride within those lines as well. And you want to make sure that you replace broken or clogged emitters. That's extremely important. You can tell when something is wrong. You'll usually see a little puddle of water beneath an emitter, or you're walking down the grove and you can actually hear it. So if you start hearing stuff coming from your sprinkler system, most likely it's going to be clogged. You also want to check lines for chewed lines from coyotes. I know in Northern County, San Diego, we've been having a lot of problems with that. So we want to make sure that you guys check on that, because if you just assume your lines are nice and clear, there's nothing wrong with them, and you're preparing for a heat wave and you end up having all these problems because you know, something is wrong, you want to make sure that you have all your basics covered. And we highly suggest that you do a measured distribution uniformity test. A rule of thumb is you want to take a minimum of 12 emitters per block or 100 meters and go ahead and test them out to make sure that they are coming out about the same. You also want to watch out if you lose a tree or a few trees within a block and you want to replace them with a younger tree, you need to make sure that you change the emitter on the younger trees, because they do not need as much water as the older trees do. So please don't forget about that as well. And, you know, that we do have some tools, you know, for especially when the younger trees, if this is their first summer going into, you want to make sure that their mulch is up to par for what's going to be happening this summer. Some people use tensiometers as well. We usually use them in avocados quite a bit. I have seen some growers also use them with their citrus as well. And just to remind you folks that we will be hosting water management workshops. I highly suggest that you guys attend. It is in person. There is a $50 fee. It is in joint with the Citrus Research Board, as well as Danelli Sakara and Khalid Bali from Davis. And we will have a lineup of speakers that will be talking about water budgeting, soil and plant relations, irrigation strategies. This is the ad for the one that will be held in the Coachella Desert. It will be at the Coachella Valley Water District tomorrow. I believe we are capping registration at 60. As of yesterday or the day before, there were still spots available. The link will close down if there is no more room. Please feel free to contact me or anybody else that is on the flyer if you have any questions. And then here is the advertisement for the exact same workshop in San Diego County. This one will take place at the San Diego County Farm Bureau. There is also a $50 fee. You register online. This one is also capped. I do not know what the numbers are as of yesterday or even today. This one will be on Thursday, June 23rd, and it will be pretty much an all-day in-person event. So I would highly suggest that you guys attend one of these. Be prepared for the summer. Irrigation is extremely important. I know in San Diego and South Riverside County, we're starting to have some problems with that, but we hope to see you guys there. And that's all I have really quickly. Thank you, Joey.
Thank you, Sonia, for sharing that information with us today. As a reminder, we're going to ask all questions at the very end of the webinar. But go ahead and please welcome today's speaker, Dr. Jim Adaskaveg, Professor of Plant Pathology at the University of California, Riverside. Dr. Adaskavege's research program focuses on the biology, epidemiology, and management of plant pathogenic fungi and bacteria of fruit and nut crops in California. For over 30 years, Dr. Adaskaveg has helped in developing most of the currently registered fungicides for fruit and nut crops in the US, as well as around the world, and continues to develop packing house methods for fungicide sensitivity and resistance monitoring, and developing international protocols with commodity and regulatory officials to open and maintain export markets for California commodities. Today, Dr. Adaskaveg will provide an overview of sweet orange scab and citrus canker and their potential impacts on citrus production in California. Jim, please go ahead and share your screen to begin the presentation.
Well, thank you very much, Joey. Appreciate the conversation here and the opportunity to talk to you about some of our research on these subjects, as well as some of our basic information of what's happening around the United States. As Joey indicated, these are not common diseases in California, and it's Elsinoe, the sweet orange scab, has only been detected recently. And we don't have citrus canker present in California, but it is a threat. And CRB asked me to talk about it as a potential invasive species. The sweet orange scab, I've been involved with CDFA. They've been involved with doing statewide surveys and working with CRB, CCQC, and other researchers at UC Davis, like Neil McRoberts. We've met and talked about these issues, and what I'm going to try to do is provide a summary of some of this information for you today. So my talk is divided into three parts. The first part is sweet orange scab. The second part is the citrus canker. And the third part is basically a review of questions that were sent out prior to the meeting. So sweet orange scab is a fungal disease, and some of the symptoms are being shown here. You can get leaf symptoms on the bottom and the upper surfaces of the leaf, and the pathogen is known as Elsinoe australis. It's basically typical symptoms commonly occur on fruit, but they also can occur on leaves and stems under ideal conditions for disease development. The fungus produces a spore case inside these scabs called a servulus. And fortunately in California, we're only dealing with the asexual stage of the organism, although the sexual stage or ascostroma of this Elsinoe has been found in Brazil. To date, nobody has found outside of Brazil has found the sexual state, and pretty much it's this anamorphic state developing in these scab lesions. So under high rainfall, the lesions become very scab-like, raised, and can coalesce into larger, as you see in the lower left, larger lesions that can deform the fruit and cause serious cosmetic effects. You can see along the bottom close-ups of these scab lesions affecting the epidermis of the fruit. And again, in those scabs, there's basically the fungus growing as mycelium, and you can see a culture of that organism growing on this specialized orange media, that round petri dish in the center of the slide. And what the organism does is it starts producing spores, and basically two types of spores are formed: a yeast-like stage shown on the midsection to the right, and then a spindle-shaped spore shown in the middle and to the left of the three images in the center of the slide. So it has two spore types, and this is kind of important information as we discuss this disease. So there are look-alike diseases to scab, sweet orange scab, and one of them is called common scab. And this is also a fungal disease caused by another Elsinoe species called Elsinoe fawcettii. This species is commonly found in Florida. The lesion can be single or irregularly grouped, and usually has these more wart-like growths, usually becoming covered with a scabby, corky tissue. Again, they're very similar between the two, but again, we'll talk about the host range, and some of the leaf symptoms there are slightly different. Another disease that we're going to talk about today is this citrus canker, and it's a bacterial disease, and it's similar to causing scabby-like lesions on fruit and leaves, but it has some distinct characteristics. The center of the circular lesions are usually brown. They are rapid lesions raised lesions surrounded by oily outline or water-soaked tissue. And then on the very outskirts, there's this yellow halo or yellow ring. So on the bottom right, you can see a close-up of these symptoms. And although it looks a little bit like scab, the combination of the oily ring, the yellow halo, and the raised corky brown lesion in the center is very distinguished for citrus canker. So when we look at the host of the sweet orange scab, the Elsinoe australis, it's basically named for the host it occurred on. So the common name, sweet orange scab, meaning basically it likes to mostly occur on sweet orange. But it can be, that's where it's the most severe. It can be found on tolerant species such as sour oranges and hybrids to a lesser extent, with moderate symptoms showing on grapefruit, lemon, mandarin, but again, mostly fruit, and occasionally or less frequently on leaves and stems. The critical time for susceptibility for fruit to become infected is during the early stages of flush in 8 weeks after bloom. In that time period, there's developing fruit. And at that particular stage of the fruit development, it's very susceptible to infection. The pathogen is carried on infected nursery stock. And it can be a pathogen on ornamental citrus plants and fruits. So it's not just occurring on cultivated citrus, it could be on other types of citrus-related plants. The conidia of the pathogen are dispersed by wind-driven rain, and insects also can spread spores of the pathogen. So the pathogen itself, as I mentioned earlier, ascospores are not known to be involved, but may contribute to survival and dispersal. Conidia are short-lived, only about a really short time period, less than a half hour at 50% humidity. So they're not long-lived, which is, again, very important from understanding the epidemiology of the disease. And these fungi are very hard to grow in culture and learn and study. And so a lot of the information what you see in asterisks here, some of that information is based on the closely related species of common scab or Elsinoe fawcettii. The mycelium is also very slow growing, and they can germinate and infect under actual free water wetness 3 to 4 hours. They can germinate and infect in a very short period of time. The yeast-like phase can reproduce by budding, and that's those smaller spores I was showing you earlier. And these things may be epiphytic on citrus and other plants, but this needs to be explored. And what I mean by epiphytic is just growing on the surface without causing disease. It's and many yeast-like organisms can be found on plant surfaces, and these yeasts are just simple fungi that bud and produce colonies of individual cells. So if we look at the disease cycle for Elsinoe, which includes Elsinoe fawcettii and Elsinoe australis, it can occur on fruits, twigs, and leaves, as it's known to develop in the United States. Again, the typical symptoms are these corky lesions with a rampant scab pustules on the fruit. And on the upper part of middle of the slide, you can see some of the oranges, and when they're mature, they get infected when they're smaller in these 8 weeks after the initial bloom period. And then they continue to develop as the fruit develops, and you can see these large scab-like lesions on the surface. Some infections can occur on stems and leaves, as also you see those raised scab-like lesions forming on the leaf margins and in the leaf petiole itself, and on the leaf blade itself. So it really needs this splashing water to move the spores from the acervulus. They disseminate with a little bit of wind movement while it's raining, and these spores can then land on new tissue, and that's basically how it's spreading from one infection plant to another healthy tissue within the same tree or within adjacent tree. The organism, because of these short-lived spores, as we were talking about, doesn't move very fast, and so it on its own, with rain and wind, it can move usually from one tree to the next is usually what happens over the course of a season or two. So the infections are very quick under those conditions, 3 to 4 hours, as I mentioned, at ideal temperatures between 70 and 80 degrees Fahrenheit. There's some differences here between Elsinoe australis. Australis only produces hyaline spores. This just means there's no pigmentation to them. And the spindle-shaped spores are thought to be the main infective propagules. Fawcettii has clear hyaline and pigmented spores, and this is a minor detail, but it is important to recognize that there are differences between these fungi. So again, the leaf and fruit susceptibility is usually 6 to 8 weeks after leaf flush. And again, the sexual stage is unknown in the United States, but has been found in Brazil. Now, some of the work that was done with surveys in California have shown that sweet orange scab was found. And the symptoms really differ from what we see in high humidity locations. Like here, the symptoms in South America of sweet orange scab shows up as these scabby-like lesions all over the fruit that kind of coalesce. In California, under low humidity, we can see that these lesions look like abrasion damage from maybe wind scarring as the fruit are moving through windy areas, moving around and brushing up against leaves or other stems. And we see this kind of symptom developing is kind of atypical from what we compare to what we see under high humidity in other parts of the United States or in other parts of the world. Again, so with these surveys, CDFA is taking these samples and they're cutting up sections of these scabs, and then they're looking for extracting DNA and trying to amplify that DNA to show that this is the pathogen. So what we do is we take these symptoms. These are provided by CDFA. You have this mud cake symptom, which is also common with wind scarring, and we can isolate from that. This is another fruit with the symptoms at the early stage of green fruit development. And we have what's been published is that there are this EAUT5 is a species-specific primer, and so this sequence of the primer DNA, we can actually amplify specifically the species of Elsinoe australis. And so what we see in the gel here is that the band here is the positive ones for SOS, and the bands that we see for the control are showing that the DNA that they extracted was present for all the samples and only in this case 4 of these samples were positive for Elsinoe australis. The reason why we're using molecular methods is because the fungus is very difficult to culture, and so this is primarily how researchers and regulating bodies around the country are currently identifying sweet orange scab. Now, the history of SOS is that in the US, this is a relatively new disease for the United States. It was found in the mid-2010 in Texas, but then was found in Louisiana, Mississippi, and then in late 2010, it was found in Florida and Arizona. This triggered statewide federal quarantines starting in 2010, and these things were then under regulation of the USDA APHIS following the procedures for a quarantine pest. CDFA surveys began in 2013, and that was the first year that it was detected in California. Subsequent surveys in 2016, 2020, and 2021, in these surveys, they were inspecting about 10% of each grove starting in the center. They collected scarred fruit prior to washing for different grower lots in packing houses, and then they collected in commercial and residential areas. And so the map I'm showing you here are just basically indicating the number of detections and what county they're occurring in. Initially in 2013, there was a find in Pomona, in Los Angeles County, then in Riverside County out in Blythe, and then down in Imperial and the Winter Haven, Calipatria areas of that county. In 2016, again, another survey was done and found more in Winter Haven. We also, CDFA did surveys in 2020 and finding both in Orange County in Tustin and in Blythe again. And then in 2021, there was 2 detections found in Riverside, the city of Riverside in Riverside County. So we note that there's ways of detecting it, and we can differentiate between Elsinoe australis, the sweet orange pathogen, versus the common scab or Elsinoe fawcettii. So we use these different primers, and here's the gels showing that fawcettii can be differentiated from australis. There's no band in the upper left for australis, and this basically says that there's no, this is negative for australis, but the primers that are available for fawcettii, which are labeled EFAW 1 or 2, are indicating that we can detect fawcettii and differentiate it from australis. Similarly, with the detection of australis with the primer, we have this E5, and basically we can detect australis. And there's 2 pathotypes, and this is again differentiating pathotype, meaning differentiating the pathogenicity on different hosts. And so citrus noxididii is part of the australis group, and it tests positive for those 2 pathotypes that are known for Elsinoe australis. And researchers have gone out and made species-specific primers for the noxididii pathotype. And again, this is how we can differentiate it from the Elsinoe australis, which is the sweet orange pathotype. And so using this primer, we get positive for all the australis, but then using the EA NAT-2 primer, we can separate it from the noxididii pathotype. So the 3 sets of primers, we can differentiate the different species and pathotypes of Elsinoe australis on citrus. So the host plants for Elsinoe pathogens, here we have australis on the left, and we have fawcettii on the right. You can see the host preference sweet orange and not so die orange are and shown in orange, so they're the most susceptible. Moderate susceptibility is grapefruit, lemons, and limes, kumquats, finger limes, and satsumas. And then the least susceptible are the sour orange, tangor, and jojoba. But jojoba that's a pathotype that has been reported from Japan, but that this pathotype does not infect sweet orange. So this is an outlier, and that's why we have that in the as a footnote. The Elsinoe fawcettii again prefers grapefruit, lemon, tangor, as well as sour orange, and then sweet orange is less susceptible compared to these other hosts to the fawcettii or common scab. So the reports here have indicated that from the surveys that I showed you earlier, these commercial citrus groves, I should say, starting in 2013 to 2016, they were found on lemon in Tangelo, and again in these locations. And the total number of detections in each year of these surveys are shown here in given totals by year. That 2011 were detected in this on the lemon host, 3 on the Tangelo, 3 on orange with some of these later surveys, and then 1 was not specified. So the detection currently stands at 18 total detections in the state of California, and 5 of these detections of the 8 were a second sample in the orchard. So, and they're going back in the CDFA surveys, going back to see if these things are moving or spreading. And so sometimes some of these things are being redundant detections, but still showing the persistence of the detections in those locations. So the impacts of detections from a CDFA, a regulatory perspective, is basically, it started in 2013 in Riverside and Imperial, Los Angeles counties. And these became regulated areas by CDFA. And they basically set up a 5-mile perimeter around these detections, a 5-mile radius, I should say, around these detections to help prevent the spread of this, because it's basically a standard procedure for responding to a quarantine pest. In 2021, APHIS has requested the federal USDA APHIS Animal Plant Health Inspection Service. They came to the state of California to develop and asked them to develop official regulations, which are quarantines, to prevent interstate spread of the pathogen. And these other detections in these other states were automatically statewide. Because California is so large and the detection areas were limited to Southern California, we basically went with CDFA's process of calling these regulated areas. So some of the restrictions that were immediately in place were that harvested fruit should be free of stems and leaves during transport. So if the pathogen's causing leaf infections and stems, if you harvest the fruit, it goes into bins. These things could be basically fly away or fly off the truck and would help disseminate the pathogen if these lesions are producing the conidia of the pathogen. And then we started asking questions about, well, we're concerned with HLB and ACP spread, and so the current regulations for that quarantine disease is tarping of the bins. So we requested that we could do either remove the stems and leaves from the bins, the harvest bins, which is quite laborious, or just providing a tarping similar to the ACP and HLB requirements for tarping the bins during transportation to the field, to the packing house. And this was approved this past spring, 2022. So this alleviates the extra burden of doing this work of removal, and the tarping should prevent the spread of the leaves and stems outside of during transportation. In the packing houses are required to wash, brush, treat the fruit with a disinfectant, treat the fruit with fungicide, and treat the fruit with fruit coating. All these steps are considered effective, but all are required. And so we were wondering about the redundancy of requiring all these steps. Regulations for limited interstate movement of nursery stock. This requires compliance with APHIS and a pre-shipment fungicide treatment and regular inspections of the nursery. More stringent requirements for nurseries when they're shipping their products to other citrus producing states. And basically the regulations are requiring approved structures with a solid roof, again, to prevent rain splash from disseminating the potential pathogen SOS to other plants, irrigation practices that reduce water splash on plant tissue, and propagation of only certified clean stock. So obviously, from a grower's point of view, you don't want to use overhead irrigation or things that are going to spread, causing splashing and moving of the spores from lesions to other fruit, healthy fruit. And again, when growers are planting an orchard using only certified propagation material, fungicides are known to be very effective against sweet orange scab. They provide barriers to the protecting the fruit, preventing the spores from germinating, and they're basically applied during this very susceptible growing period from flush to 8 weeks in other locations. And the result is reducing the amount of total inoculum and reducing the spread of the disease. And then I have a question here that when maybe in our case where we've detected these SOS findings in certain locations, fungicides could be applied, and this would be the next logical step to prevent the spread of SOS. The fungus attacks, as far as timing, we went over this already, but the fungus attacks mostly young fruit susceptible up to about 6 to 8 weeks after petal fall. Management is based in other locations on 2 fungicide applications, one at 2/3 petal fall and a second about 2 to 3 weeks later. And then again, as we talked about, once you find these symptoms and fruit infected fruit, you don't want to remove them from the orchard without specific procedures to prevent the spread under the quarantine rules. Leaves and stems should be removed, or tarping should be required and is required for if you're going to harvest that fruit. These types of symptoms can be also sorted if they're obscure or not well detected. They can be further sorted in the packing house to remove those potential fruit with infections. So packing houses can, if they see symptoms of scab or wind scarring, those fruit can be sorted and removed during that packing process. So we go back to the federal order for packing house procedures. And again, this is for citrus and Fortunella species, and under the SOS quarantine area or regulated area, which will soon become an official quarantine zone. To again, this is basically to move fruit within the state with certified approved methods and under the conditions. So washing, brushing, these are all required steps if you're going to be packing fruit from a quarantine or regulated area. The surface disinfectant with this is often confused with one of the following. So in other words, you have to have a surface disinfectant, but you could use various materials like sodium hypochlorite, you could use sodium orthophenylphenate, and you could use PAA or peroxyacetic acid. The concentrations are shown there for sodium hypochlorite, 200 parts per million. For sodium orthophenylphenate, 1.86 to 2%. And for PAA, about 85 parts per million. And the times for each are also shown, from ranging from 45 seconds to 1 minute in length of exposure to these surface. But you don't have to do all of those. You have to use one of them. With the effects, with the cost and disposal costs of sodium orthophenylphenate, this is an option that's least likely to be followed or suggested to be used because of the cost, again, in disposal. So you're left with basically the PAA or the sodium hypochlorite, which are fairly inexpensive standard procedures for citrus packing houses. They're treated with fungicide at a time of packing. Again, this could be a Maslow, TBZ, or some combination like Graduate A+. And then the fruit also has to be waxed or used with a fruit coating. And sometimes the fungicides are applied in the fruit coating. So both of those steps could be done at the same time. But 1 through 5, all those steps are considered highly effective. And those things have to be followed for packers that are packing fruit from a quarantine or regulated area. The biological characteristics of Elsinoe is that they are produces these hyaline conidia only, which are short-lived. They require water for production, dispersal, survival, and germination of the spores. Wind speeds of about 4.5 miles per hour are required to liberate. It's a little bit more than a gentle breeze, but it's required, wind gusts to liberate the conidia from these spore masses in those scabs. And as little as 2.5 to 3.5 hours of wetness under ideal temperatures around 70 degrees. The conidia are short-lived. They're hyaline, so they're subject to UV destruction. They're short-lived, and they're basically can be desiccated when typical 50% relative humidity or lower conditions can shorten their lifespan to just a half hour. So implication scab epidemics are caused by abundant water dispersed hyaline that have the greatest cause of potential within the canopy of the tree. Opportunity to move to another orchard or another grove is very low unless they're planted right up against each other. So we're most likely to see movement within the tree, followed by tree to tree, and eventually, if it keeps spreading, eventually from one grove to an adjacent grove. So the 5-mile radius has been identified as a restricted area, and it seems to be very strict interpretation of this based on the biology. And we've proposed reducing that 5-mile radius, but this is still under consideration. Now I'm going to go to the second part of the talk real quickly. Citrus bacterial canker. This is basically caused by 2 types of organisms, Asiatic canker and South American canker. As you see, the Asiatic canker is this bacterial disease caused by Xanthomonas citri, Pathovar citri. And there's synonyms where it was described previously. The South American canker is caused by Xanthomonas citri, Pathovar aurantifolii. And so the most common one is this citri worldwide, and the citri or Pathovar aurantifolia, or South American canker, is restricted to South America currently in Argentina, Brazil, Paraguay, and Uruguay. As the name implies, xantho is yellow. The culture of the organism, when you culture it, the bacterium is yellow, and it has a rod shaped with polar flagella, so it's able to swim in films of water. The symptoms, again, of canker, they have that raised corky lesion with a oily ring followed by a chlorotic or yellow halo. And so these are characteristic symptoms both on leaves and fruit of the symptoms of canker. And basically we call it citrus canker because we only have the Asiatic canker in the United States. So the taxonomy has changed. If you're those of you looking at literature, you'll see that the name has been changed as we learn more and more about these organisms. And these are the current names that I introduced to you in the previous slide. The distribution of the Asiatic canker is established in various citrus producing areas from Southeast Asia, Africa, Australia, Brazil, the Middle East, and in the United States. In addition to Florida, there have been finds in Louisiana and Texas in 2014 and 2016, and the disease is not known to occur in California. So this is, again, the take-home message here is that it has the potential to be an invasive species. And one of the things that makes California unique is that we don't have all these quarantine pests. And so this is very important that we understand this disease so we can prevent its invasiveness into California. There's no record of seed transmission, and if there is going to be an outbreak in California, it's most likely going to occur from improper movement of host material. And so using only certified plant material is very important for the whole industry. So the disease cycle of citrus canker, the bacterium enters the plant through natural openings like stomata and multiplies and grows onto leaves, stems, and fruit, as we just saw in the symptoms. So we can pick it up here as the windblown rain carries inoculum that oozes out of the lesions. And then on water films, the organism can then grow and swim, essentially multiply and swim in these water films through the stomata and get into the internal portion of the leaf. Other injuries like hedging or any kinds of injuries, the organism is going to take advantage of that as well. So wherever there's wetness, the bacterium can be released, and then it can be dispersed, and it can then get into the plant, and it can be blowing in the wind and from rain splash. It's not considered an epiphytic bacterium, but it can survive on the plant surface for a few days in what we call biofilms or organized aggregates. And so as we get more rain, bacteria ooze out, and then the splashingness will carry it to the stems and leaves and the fruit. So critical environmental conditions for infection is the temperature and leaf wetness duration are key factors. Young leaves of sweet orange are most susceptible. Temperatures of 12 and 40 C are necessary for symptoms to develop, and optimum is somewhere between 25 and 35. So this is the overall range, but this 25 to 35 centigrade, this would be 75 to about 80 or 90 to 88 or 90 degrees. So short duration rainfalls favor the excudation, meaning that out of these lesions, the bacteria is oozed out. The the severity is directly related to how long wetness duration and the frequency of rains. And the bacteria may stay latent in the plant for a few days to a week before you actually start seeing symptoms. So you can go through these infection periods. The most critical period for fruit infection is following using a pressurized sprayer is 60 to 90 days after fruit set. So that's the most susceptible period of the green fruit. Again, we talked about the different forms of citri, Xanthomonas citri. There's the Asiatic canker, which is Pathovar citri. There's two strains known, the A and the A prime or A star. And this is because there's overlap with Citrus aurantifolia with the A star also infecting aurantifolia, which is the same as the Pathovar aurantifolia. And these strains are basically called B and C. So that's basically the taxonomy and the host preference of these species. The capacity to spread is natural dispersion, as we've talked about, occurs through rain and weather events such as thunderstorms or hurricanes, which are not unheard of in California, typically can occur down in the Imperial area with summer monsoons. These would be how we can get infection and spread once it's introduced. The big problem is human activities can favor the spread of the movement through contaminated plant material. And again, shipping from nurseries that are infected into California would be the long distance dispersion of the pathogen. And because it's a bacterium, it can be transmitted through machinery, clothing. If you're going into wet orchards that have symptoms, tools can be contaminated. These are very similar to other bacterial diseases. The bacteria can form films on those tools. And then if you're using them for pruning, you could be actually infecting your plants. Another unfortunate aspects that can occur in residential areas, parks and so forth, gardens, and then this increases the likelihood of establishment of the organism. So, it's very important to keep that from being introduced. Once it's introduced into an area, it can spread on an average of 30 meters to 830 meters. And again, this is what has been measured in Florida, Argentina, and Australia, just by natural rainfall moving the organism from one tree to the other. So the risk factors, a risk factor is both biotic and abiotic factors that can increase the probability of infestation. And so what we're looking at is commercial shipments is the biggest threat of contaminated fruit or contaminated plant material bringing into the state. Once you're handling these things, these are high risk for fruit injuries, and you can get more infection. The higher risk areas can be defined as the epidemiological units contiguous to the risk locations. So once you process, if in the packing house, if you process these fruit that have cankers and then you put it out in a coal pile, and it's near an orchard, this can be considered a high risk option for moving the disease from one location to the other. And again, the most likely place of movement would be through budwood, rootstock, seedlings, handling of plant material, even ornamental citrus related plants can all be possible ways that this organism can be introduced. And lastly, once you have citrus leaf miner, which is common in California, you can exacerbate the canker disease as shown in Florida and Brazil.
Now we're going to go ahead and ask the questions in the chat. Looks like we have one question in here. If you have any questions for Jim, please add them to the Q&A section and we'll get those to him. But the first one we have, did the sweet orange scab surveys include the San Joaquin Valley?
There are statewide surveys on citrus, so it did include San Joaquin Valley, but the focus has been in the detections down in Southern California. But they do these statewide citrus surveys, CDFA. They don't go to every citrus orchard, but they target certain ones. I don't know exactly how they choose which orchard, but they go to different parts of these citrus producing areas, whether it's coastal or desert or in the Central Valley. And so to date, from these statewide surveys, it's only been found in the counties I listed in Southern California. I guess we have a comment from Victoria. She's the CDFA person in charge, and they look for canker and SOS in these surveys, and as well as multi-pest surveys. And they are conducting SOS delimitation around areas where SOS has been detected in the past.
So we have another question from Scott Carlisle. Do the SOS packing house treatments need to occur within the quarantine zone?
The packing house treatments, if you're receiving fruit from a quarantine zone, you have to, or you're packing fruit in the quarantine zone, you have to follow those procedures. Yes.
And will SOS and citrus canker be able to survive in the San Joaquin Valley because we get temperatures over 100 for weeks on end and low humidity?
Well, there are other Xanthomonas species living in the Central Valley, and this Xanthomonas species is a warm, it comes from the tropics, so it's a warm temperature pathogen. And so I think it can survive even very high temperatures in the Central Valley if it was introduced.
All right, those are all the questions we have for now. If anyone has any additional questions, please put them in the chat. Oh, here's a clarification on Scott Carlisle's question. He said, can I ship fruit from the SOS quarantine zone to be processed at a house outside of the quarantine zone?
Well, I think we'll have if Victoria's on the line, we could have her try to address that. Victoria, if you do have a comment on that, I'd have to check on the regulations for that specific question.
I would like to thank Sonia and Jim for sharing this information with us today, and our participants for joining the Citrus Research Board for today's webinar. CRB is actively planning for our annual California Citrus Conference to be held October 19th, 2022 at the Whiteside Convention Center. We hope to see everyone at the conference and will provide further information in the coming months. Please make sure to mark your calendar for our final webinar next Tuesday with Dr. Mike Bruce and Dr. Tracy Kahn, who will be discussing rootstock characteristics and new scions in California. We will stick around for a few minutes and try to answer any additional questions, but if anyone needs to hop off, you're more than welcome to do so now. So Victoria said that a packer would have to have a compliance agreement to accept fruit from an SOS area and would have to conduct the USDA approved packing procedure. And then we have another question from Tracy. Can SOS lead to overall decline in a tree?
That's under these conditions in California, that's yet to be seen, but in other places where there's high humidity, yes, it can cause severe fruit infections, twig infections, leaf infections, and again, on those varieties that are preferred by the different pathotypes. Right now, I would say that in California, the symptoms are relatively limited to fruit, but it potentially can occur on these other tissues. So I don't know if it will cause severe dieback and eventual overall decline. It does diminish the fruit quality, and those have to be graded out, even the wind scar symptoms of scab. So this would be something that would directly economically affect the production of citrus. As far as the overall decline, specifically for California, I don't think at this point we know that it's going to lead to that, but in other parts of the world, it gets so severe that the tree becomes uneconomical.
So Andrea asked, the diseases can survive low humidity in the San Joaquin Valley?
Well, apparently, out in the Imperial and the eastern part of Riverside, it's very low humidity. Even in, all through where the other detections were found in the city of Riverside or over in Tustin and these locations, it's very similar to the Central Valley with low humidity in most of the year. So these organisms, once they get established, I think it's difficult to say that just an environmental condition like lower humidity would result in the decline of that organism. So again, it's important to follow these procedures to prevent the spread of these types of pathogens like SOS. And overall, some of the information I think is that these organisms, we never thought it would come to California being such a drier citrus production region, but again, here it is to our amazement. So this is what we're finding.
Well, thank you, Jim. It looks like that's the last question we have for now, but like I said, we'll keep the Zoom open if anyone has any technical questions. And we can provide Jim or Sonia's contact information if someone has additional questions they'd like to follow up on. But thank you everyone for attending, and we hope to see you next week for Tracy and Mike's presentation.
Thank you again, CRB, and we'll be in touch.